Hello everyone, I'm Nagao from Okayama University. This presentation is about my recent BBA paper. The title is Excitation Energy Transfer in Heterocysts Isolated from the Cyanobacterium Anabaena PCC 7120 as studied by time-resolved fluorescence spectroscopy. The authors are shown here. So collaborated with Professor Shen and Ehira and Akimoto and others. So this presentation has no figure and tables because of copyright of BBA publishers. So please download this PDF file and watch this presentation during checking the PDF result. Okay, so let's get started. Okay, so summary for this presentation. So we examine the excitation energy transfer mechanisms in heterocysts by time-resolved fluorescence spectroscopy. So the main topics is a characterization of heterocysts. So the 4 bullet point is here. The absence of PS2 retaining manganese calcium cluster by analysis of oxygen evolving activity and some luminescence. And second is the absence of PS2 dimer and monomer by two-dimensional BNSD page analysis in heterocyst. The third is observation of fluorescence originating from assembly intermediate of PS2, namely a PS2 reaction center complex, so D1, D2, B559 alpha beta subunit, and PSPI, so reaction center complex. The first is not transferring energy from PS2 to PS1, so namely not occurring a spillover in this heterocyst samples. So first of all, about heterocysts. So heterocysts are formed in some cyanobacteria group under nitrogen deficient conditions. The heterocysts perform nitrogen fixations, converting nitrogen gas to ammonia. So Anabaena PCC 7120, hereafter referred to as Anabaena, is one of the filamentous cyanobacteria. So red arrows are heterocysts. So this one and this one, this, and the other cells is a vegetative cells. So there are a lot of subjects of heterocyst photosynthesis. First, it is difficult to prepare high purification of heterocyst. Second, there are a few studies of photosynthetic properties of heterocyst. Third, it is thought same photosynthetic properties among heterocysts irrespective of different species. Fourth, there is no study about excitation energy dynamics of pigment-protein complex in this Anabaena heterocyst. In this study, we examine the excitation energy transfer processes of pigment-protein complexes in Anabaena heterocyst. Okay, this is figure 1. So figure 1 shows PS2 activities, so namely oxygen evolving activities and thermoluminescence. So figure 1a is oxygen evolving activities. So vegetative thylakoid showed 95 activity of oxygen evolving activities. However, heterocyst thylakoids showed not detectable levels of oxygen evolving activities. This suggests heterocyst does not proceed this Koch cycle. So figure 1b shows some luminescence, so TL glow curve. vegetative sarcoid showed a Q band originating from S2QA charge recombination MPS2. However, this band does not occur in heterocyst sarcoid. This strongly reflects the S1-S2 transitions does not proceed in heterocyst. So in other words, heterocyst does not have manganese calcium clusters. Okay, figure 2a shows Western blotting analysis of heterocyst and vegetative sarcoid using the PSAAB and D1. So cross-react band appeared in vegetative and heterocyst sarcoid using a PSAAB antibody. However, D1 band is less extent in heterocyst sarcoid compared with vegetative sarcoid. And figure 2bc shows BN page and two-dimensional BN SDS page. And first PS1 tetramer and dimer and monomer can be detected in heterocyst. However, PS dimer and monomer band was not observed in heterocyst sarcoid. So these results suggest the assembly intermediate of PS2 exists in heterocyst sarcoid. Figure 3a shows absorption spectra of the two samples under the liquid nitrogen condition. You can see similar absorption spectra of the two samples, especially in the wavelength region of chlorophyll A QY. Figure 3b showed fluorescence emission spectra of the two samples under the liquid nitrogen condition. Vegetative sarcoid showed characteristic fluorescence at around 685 nanometer and 695 nanometer. These two bands originate from CP43 and CP47 in PS2 complex. However, the heterocyst sarcoid showed no these two band, but appear at around 683 nanometer. So this band, the 683 nanometer fluorescence band may be due to the chlorophyll component from a PS2RC complex. So PS2RC may exist in heterocyst. This slide showed unique feature of PS2 assembly intermediate. Following steps for PS2 assembly. The first is a PS2 RC complex consisting of D1, D2, cytochrome B559, and PSBI, the smallest complex in PS2. Second is a complex of PS2 RC with a CP47 and several small subunits. And third is a PS2 monomer. This complex can evolve oxygen, so can perform a water oxidation reaction. So 4 complex. And finally, a PS2 dimer was assembled. So these characteristic complex showed a unique fluorescence peak under the liquid nitrogen conditions. So PS2RC showed a peak at around 683 nanometer, and PS2RC CP47 showed a peak at around 688 nanometer. And PS2 monomer and dimer showed 2 peaks at around 685 and 695 nanometers. And also these two complexes of PS2RC and PS2RC CP47 does not perform water oxidation reaction because this complex does not have manganese calcium cluster. However, these two complexes can perform a water oxidation reaction. So figure 4 showed time resolved fluorescence spectra of the two samples under the liquid nitrogen condition, so TRFS, showed similar PS1 fluorescence between the two samples. The vegetative sarcoid showed fluorescence band 685 697 in the time range of 0 to 110 picosecond just after excitation. However, the heterocyst sarcoid showed almost no F685, but strong F697 originating from PS1 fluorescence just after excitation. And also the heterocyst sarcoid showed F683 in the time range of a few nanoseconds. Figure 5 showed the fluorescence decay associated spectra. So FDAS showed similar spectral shape in PS1 region between the two samples. However, the heterocyst sarcoid showed no delayed fluorescence at 22 nanosecond. So this suggests the spillover does not occur in the heterocyst. So spillover means excitation energy transfer from PS2 to PS1. Okay, this is a summary. So in this presentation, I showed the photosynthetic property in heterocyst from Anabaena SPPCC7120. The characteristics are shown here. So almost no difference of PS1 property at protein level and spectroscopic levels. And that raised the existence of PS2RC in heterocyst. So this complex was not detected by two-dimensional BNSDS page. However, this complex can be detected by fluorescence spectroscopy. And also, spillover does not occur between a PS2RC and a PS1. Thank you for your attention.